Hi, this video is to go over some of the material related to plant nutrition that I wasn't able to cover in class, and also to add some interesting material and some material that will help you complete the objectives. So the first topic is phytoremediation, and what this really is the exploitation of nutrient uptake pathways that are present in plants, and to use these uptake pathways to detoxify soils that have been made toxic by industrial processes. Here's one example. So these plants are grown in a media that contains mercury, and mercury is a neurotoxin for humans. It's also toxic to plants, and you can see that here. These wild-type plants fail to germinate and grow. Whereas on the other hand, these big green plants on the side here, these are transgenic. They've been genetically engineered to have high expression of a protein that is a mercury chaperone. And what that means is that it binds to mercury that makes its way inside of the plant cells. So that allows the plants to grow, but it also takes the mercury up out of the soil and makes it slightly easier to dispose of. Here's another example. This is the Badger ammunition plant that was built in Wisconsin for World War II, and it's a very toxic site because of all of the explosives that have leached into the soil. One of them is TNT. In the drawing here you see that TNT has a lot of nitrogen on it, and plants need nitrogen. Down here these are two plants. The transgenic plant has been engineered to use TNT as a nitrogen source. So when these plants grow in that soil, they break down the TNT and use it as their nitrogen source. That also helps to detoxify the soil. Topic number 2. Did you know that plants have the internet? They do. This is called the Wood Wide Web hypothesis, and I'm going to share a little bit of information about how this was discovered. All right, so the experiment here involved totally encasing a plant in a bubble that allowed the researchers to control what gas it was exposed to. And they exposed these plants to radioactive carbon as CO2. Now, this tree here is going to fix that carbon and make it into sugars. But interestingly, when they looked at nearby plants, they found that those plants also had the radioactive carbon as well, even though they hadn't been exposed to it. So they have radioactive sugars. And what they figured out was happening, we can draw in the roots of these two. Oh, there was a fungus in the soil. And we can draw the roots in of these two plants here. And this fungus is actually making connections. It is cytoplasmically linking these two plants together and allowing sugar to move from one to the other. So these fungi in the soil are very, very common. They make these long extensions called mycelium that spread through the soil, and they can actually make very intimate connections with plant roots. All right, now here's a really cool follow-up experiment. So again, they have this tree that is exposed to radioactive carbon, and then they have some nearby plants that are in different lighting conditions, either deep shade, partial shade, or full light. And they studied how sugar moves from this tree into these, what we'll call receiver plants. And so the fungus is there in the soil, and it's linked to, it's linking the roots together of all of these plants. And what they found was that the plant in the deep shade, it got the most sugars from that plant. In other words, the fungi, these fungus in the soil, were conducting sugars to receiver plants based on their metabolic need. So the plant in the deep shade, it has the most trouble making its own sugar, and so it actually gets the most sugar exported to it by this fungus. So the idea here is that this fungus is kind of keeping its metabolic network healthy, and that not just sugars, but probably other important plant nutrients are able to move from plant to plant through these fungus. This is another example of a symbiotic relationship between the fungus and plant.